In this lecture, we'll be discussing the citric acid cycle, and this is the next step in metabolism after glycolysis. And as you can see from the title slide, it is a cycle, and because of that, it's a circle, so it doesn't really have a beginning or an end technically, but we really consider the step where acetyl-CoA goes into this cycle as the first step. So that's really our reference point. And how did we get to acetyl-CoA? There is actually one more step after glycolysis to convert pyruvate into acetyl-CoA. Pyruvate has 3 carbons, acetyl-CoA has 2, so we lose a carbon in this step to get it ready for the citric acid cycle. Okay, I like this figure on the title slide because it shows you how many carbons each of these compounds, intermediates in the citric acid cycle have, which I think is helpful in keeping track of the carbons, because in the process of doing a cycle, we're going to lose 2 carbons as CO2, and we bring 2 carbons in from acetyl-CoA. Okay, so it in that sense, it's sort of a neutral in terms of carbons. The other things that are produced by the citric acid cycle, you can see these NADHs. There are 3 of them, a GTP, and also an FADH2. So the NADHs and FADH2 are all electron carriers. They bring those electrons then to the respiratory chain. And GTP is, equivalent to ATP in terms of cellular energy. So this cycle really is responsible for quite a bit of energy production, although it might not be apparent yet. But when we cover chapter 19, it'll really make sense why this yields so much energy. Okay, so this is from chapter 16. Right. And we're going to be covering most of the topics in this chapter except the glyoxylate cycle. Right. If you look at glycolysis and how much energy is produced in terms of a delta G value, you add up all the delta G values for all the steps in glycolysis, and you get an overall negative value, but it's fairly small compared to if you took glucose and you did a combustion reaction and fully oxidized it to CO2 and water. That delta G is really large, a really large negative value. So we're only getting a small amount of the energy out of glucose by converting it into pyruvate or 2 molecules of pyruvate. So there's more energy left over in those molecules of pyruvate. Okay. And how you get that out is through cellular respiration. Respiration is the process in which cells consume oxygen and produce CO2. Okay. It provides much more energy in terms of ATP from glucose than does glycolysis. Okay, glycolysis is really, if you're thinking evolutionarily, it was sort of the first major energy producing pathway, and it can occur with or without oxygen. But as the Earth had developed an oxygen atmosphere, respiration sort of took over, and it actually, increased very significantly the amount of energy that can be produced from especially from glucose. Okay. Respiration also captures energy stored in the form of lipids and amino acids. We'll see a little bit of this amino acid, how they play into this within the citric acid cycle. You won't really be responsible for specifics of that, but we will see some amino acid metabolism, or at least allude to it. The respiration itself occurs in three major stages. Okay, one is the production of acetyl-CoA, and I should mention I pronounce it acetyl-CoA. I've heard some people call it acetyl-CoA. So whatever your preference is there, I call it acetyl-CoA. It's just easier for me. Okay. So the three stages: acetyl-CoA production. Okay, that we'll see that involves glycolysis, and then through what's known as pyruvate dehydrogenase to form acetyl-CoA. Then acetyl-CoA oxidation, which would be the citric acid cycle, and then finally electron transfer and oxidative phosphorylation. That's what we refer to as the main respiratory chain. And that will be the topic of chapter 19. Okay. So if we look at this just in a diagram form, stage one, the production of acetyl-CoA, we've talked about glycolysis, so we've done this part of it. Okay, there's some electrons that are being produced here in the form of reduced NADH. So we've taken glucose to pyruvate. Okay, today we're going to talk about one of the things. The first thing we'll talk about is this pyruvate dehydrogenase complex, which takes off a carbon from pyruvate to make CO2, and then forms acetyl-CoA with the other two carbons. Okay, that itself reduces an NADH, so that's electrons. These electrons then flow into the main respiratory chain, which would be stage 3. You also have stage 1 in terms of fatty acids and amino acids. There's ways to convert them into acetyl-CoA as well. And stage 1 generates some ATP, NADH, and FADH2. Stage 2 is where the citric acid cycle lies. Okay, acetyl-CoA goes into the citric acid cycle, which we'll see in a much greater detail. 2 carbons come out in the form of CO2, and then you have many more reduced electron carriers. And then finally, stage 3 is where those electron carriers, NADH, FADH2, go into the main respiratory chain or electron transfer chain in the mitochondrial inner membrane. And this produces lots of ATP. So this is the end goal for cellular respiration. In eukaryotes, the citric acid cycle occurs in the mitochondria. Right? And we should contrast that with glycolysis. Glycolysis occurs in the cytoplasm. Okay? Citric acid cycle is occurring in the mitochondrial matrix to be specific. There's a little note here. Technically, it's in the matrix except for one enzyme in this cycle, and that's Succinate Dehydrogenase, which is in the inner membrane of the mitochondria. And we'll see that Succinate Dehydrogenase is actually part of the respiratory chain as well. Oxidative phosphorylation, the respiratory chain, occurs in the inner membrane, right? And that's, Succinate Dehydrogenase being part of that, it is also in the inner membrane. And I should mention the figure here. This is just a basic of what's going on with the main respiratory chain. You have electrons being transferred by this chain, and what that does is it helps pump protons into the inner mitochondrial membrane space from the matrix, and then that proton gradient is then used to produce as it flows back inside with its gradient, it's used to produce ATP by ATP synthase, or a complex 5. So this is, a topic we'll cover in chapter 19. Okay. The other thing I wanted to mention too, the ultimate electron acceptor is oxygen. So that's why, you need to breathe oxygen, because you need that to accept the electrons to keep this whole system moving to produce ATP. Right? And plants are a little bit, they do have mitochondria, but they also have chloroplasts, which is a very similar system, just sort of set up in reverse. They're pumping the protons into the chloroplasts. Then they're used to produce ATP outside of the chloroplasts, but a very similar system. Electron, where they come from in the acceptor is a little bit different. We won't be really covering this in this class. You won't really be responsible for knowing that in this class. Okay, so let's talk about our next step. We left off with glycolysis, producing pyruvate. Okay. Well, to get into the citric acid cycle, as we've seen, we need to have acetyl-CoA. So how do we convert pyruvate into acetyl-CoA? Right. And that's with this very you could consider this a little bit complicated enzyme system, the pyruvate dehydrogenase complex. And it's actually a system of 3 main enzymes. Okay, there's some regulatory enzymes associated with it, but we're going to focus on the 3 main parts of it. And easily enough, they're called E1, E2, and E3, which you could sort of think of as enzyme 1, enzyme 2, enzyme 3. Okay. The net reaction is an oxidative decarboxylation of pyruvate. What does that mean? It means we're cutting off a CO2 from pyruvate. Okay, here's pyruvate. Okay, we're cutting this CO2 off. We're losing that as CO2. And then we're doing an oxidation reduction step where pyruvate will be giving up electrons and NAD plus will be reduced to NADH. Okay, these carbons lost as CO2 are going to be the first carbons of glucose to be fully oxidized. Okay. And this reaction, again, is catalyzed by this pyruvate dehydrogenase complex. Okay. This complex requires 5 coenzymes. There are 3 prosthetic groups, which we'll talk about in some detail, and 2 what you can think of as coenzymes or co-substrates. Okay, so the prosthetic groups are TPP, lipoic acid attached to a lysine, so lipoyl lysine, and FAD. Okay, and the co-substrates are NAD plus and coenzyme A. Here's a little detailed picture of what's going on with these prosthetic groups and coenzymes. So, and we'll see this again in a different form. But I like this figure because it shows you, this is what the structure of TPP is. It's sort of complicated, but really what you need to focus on is this part. This is the part that gets utilized. And what it does is basically it transfers a carbon compound. Okay. So pyruvate comes in, the enzyme cuts off CO2, and the other 2 carbon part of pyruvate is going to get stuck to the TPP. Okay, that TPP then transports those 2 carbons to lipoic acid. Okay, this lipoic acid is connected to a lysine in the enzyme, enzyme 2. So it just transfers from TPP to lipoic acid. Okay, in doing that, then lipoic acid connects these two carbons with coenzyme A, and that leaves as the product. So that's what we call acetyl-CoA. There's an acetyl group attached to coenzyme A. Okay, that leaves. In the process of doing that, lipoic acid becomes reduced, and it needs to be oxidized to be able to do this again. And so that's where we have this oxidation reduction cycle. Okay, so FAD, the oxidized cofactor FAD, will reduce, will oxidize lipoic acid back to the oxidized form. Okay, so when lipoic acid has hydrogens on it, that's when it's reduced. Those sulfhydryl groups tell you it's reduced. When you have that disulfide bond in lipoic acid, that's when you know it's oxidized. Okay. So oxidized FAD and reduced lipoic acid will do an electron transfer between each other, and you'll get oxidized lipoic acid and reduced FADH2. Okay. That's all well and good. FAD is actually connected to this enzyme, so it needs a way to recycle itself in those electrons, and it uses this NAD plus reacts, so oxidized NAD plus reacts with reduced FADH2, transfers electrons to form NADH and a proton, and reoxidizes itself so it can, function in this cycle again. And this NADH is then a free electron carrier. It can go into the main respiratory chain. Okay, so that's it, this complex in a nutshell. Okay. So let's look at some of these cofactors in a little bit more detail. Coenzyme A is a very complicated structure. If you look at it, you have an adenine base here. You have ribose 3-phosphate, so you have a sugar that has a phosphate at the 3' position. A couple phosphates, all right? So that would be sort of like a ADP, right, with this extra phosphate group on the ribose. That's connected to what's known as a pantothenic acid. Not really important that you know that structure. And then there's this what's known as mercaptoethylamine. So you have a 2-carbon with an amine group, and then the mercapto part means there's this sulfhydryl group, this thiol at the end. Okay, this whole molecule, the business end of it is this sulfhydryl. Okay, the only function of this is to accept and carry, and really for this class you can think of it as the only function is to accept and carry acetyl groups. Okay, that attaches to acetyl groups and it carries them. And you can think of this whole big thing as sort of like a handle. I've heard it described as. Kevin Ahern, who teaches at Oregon State, he describes this as a handle, and I think that's a good way to think of it. Okay, and many times you see it abbreviated as just this. You see the acetyl part drawn out, but then the coenzyme A is just abbreviated as the sulfur connected to your acetyl group, and then CoA, right? This whole part is coenzyme A. And the structure of it's really not important for us. All we care about is that it's connected through this carbon-sulfur bond to an acetyl group. Okay, what about lipoic acid, or lipolysine, as it's known when it's connected to a lysine residue? Well, it's this chain of carbons, okay, connected to a lysine. So it's a very long tether, which is important. That means it can be used to sort of move things from one place to another. Okay. And the business side, as we mentioned, is the disulfide group here. So in oxidized, there's a disulfide bond, and it's a 5-membered ring. When it's reduced, these are sulfhydryls, which then can have other molecules attached to them, one of those being this acetyl group. Okay. So the pyruvate dehydrogenase complex is extremely large. It's up to 10 million Daltons in weight, and it's multi-enzyme. Okay, and we've mentioned this just briefly, but there are 3 main enzymes involved in it: E1, E2, or E3. Okay, E1 is called pyruvate dehydrogenase, so that's a little bit confusing, right? Pyruvate dehydrogenase complex, or PDC, is what we call the whole complex. E1 of this complex is also referred to as pyruvate dehydrogenase, or PDH. Okay. So if you're talking about the complex or just the enzyme, you need to sort of specify that. Typically, you refer to it as the complex because that is what has the overall function. E2 is referred to as dihydrolipoyl transacetylase. Okay, big name. All that means is that it has a lipoic acid, and it's moving an acetyl group. Right? And then E3 is dihydrolipoyl dehydrogenase, and that dehydrogenase tells you that it's performing an oxidation reduction reaction. Okay, so E2 is what moves the acetyl group, and E3 is what it does the redox chemistry to recycle the lipoic acid. Okay, there's a couple other proteins that are sort of regulatory, and we won't really discuss them very much. We'll mention the phosphatase and kinase activities, talking about regulation, but we won't really focus too much on those. Okay, what are the advantages of having multi-enzyme complexes? Well, you have when enzymes that need to perform a function, when they're very close together in a complex, there's a very short distance between them, between their catalytic sites, so it allows a very fast movement of substrates from one site to another. It also minimizes side reactions. So if these are, very far apart and the substrates have to come off of one enzyme and find the other enzyme, the potential for side reactions would increase greatly. And then finally, the regulation of one subunit can affect the entire complex, which we'll see is utilized in the PDC. Just a picture of what this looks like. This is Cryo electron microscopy, and you can see that these complexes are very large. You can actually see them in an electron microscope, and there's something like 50 nanometers in diameter. Okay. And a picture of this, utilizing that data, if you do like a molecular space filling, would look something like this. Okay, the green core in here are the E2 subunits, and this whole complex consists of 60 separate molecules of E2. So there's 60 E2 enzymes forming this core complex in here. And it's arranged, not very important, but it's 20 trimers to form a dodecahedron. Okay, so there's a lot of structure here. Okay, it's thought that up to 30 E1 enzymes and 6 E3 enzymes are present in this complex. Okay, and these sort of green things here are tether proteins. Okay. So the ratio of E1, E2, and E3 in this complex is 30 E1s, 60 E2s, and 6 E3s. right? So that's a very, very big complex. You have a lot of copies of these enzymes close together. Okay. The ratio can change, though, reflecting the certain metabolic requirements of the tissue in question. This is I believe, crystal structure from E2. So this is just the E2 subunit crystallize out, and it forms, this very complex structure. I've tried to, in my research, I tried to purify the E2 protein, and I was unsuccessful. And that probably one of the reasons why is that you can't simply get this out by itself, the one E2 protein, because it forms such a large complex. It interacts with itself so much that it probably makes it, fairly easy to precipitate out and not be able to be purified very easily. Right, so let's look at the sequence of events in the oxidative decarboxylation of pyruvate. Okay. Enzyme 1 has the first couple steps. One is decarboxylation of pyruvate to an aldehyde, and step 2 is the oxidation of that aldehyde to a carboxylic acid. Okay, why do you do that? As we'll see, we'll see that on a different slide. Okay. The enzyme 2, E2, has the third step, and that's the formation of the Acetyl-Coa product. Enzyme 3 then does a reoxidation of the lipoic acid cofactor, and then regeneration of the oxidized FAD cofactor. Okay, so there's 2 oxidation reduction steps catalyzed by E3, and that also forms that second product of NADH. So when we look at this in a probably easier to understand form, right, this is yellow E1, the green is E2, and the reddish color is E3. Okay, so what's happening? So the first step is pyruvate attaches to TPP, and when it does that, that CO2 group on pyruvate is cleaved off. Okay. Then you have the rest of the molecule from pyruvate's attached to PPP. That's oxidized into the acetyl group when it's stuck to lipoic acid on E2. Okay, that lipolysine then carries that acetyl group to a different site. It's attached to coenzyme A at that site. Okay, that leaves as the product acetyl-CoA. In doing so, right, you have oxidized lipoic acid that's reduced, that then acts to oxidize those 2 carbons into an acetyl group. Right. Lipoic acid is then in the reduced form. It needs to be recycled back to the oxidized form, and that's what FAD is for in E3. When FADH2, the reduced form, needs to be recycled back, the E3 uses the NADH to make NAD plus, to make NADH. Okay. So this is probably one of the better pictures to look at. It kind of shows you a little bit of everything: the structures, the enzymes, what's going on, and the important cofactors and prosthetic groups. Right. If you look at the mechanism just in a little bit more detail, that's shown here, right? When you have this hydroxyethyl group from pyruvate attached to TPP, it looks like this, and you can get sort of an equilibrium here between the double bonded form and a single bonded carbanion, and in this carbanion form, that's when you get the electron nucleophilic attack to lipoic acid. Okay, then you have a rearrangement when it's attached to both TPP and lipoic acid, and that's when you get this oxidation here to make a carbonyl group. Okay. Part 2, you have coenzyme. So this would be in one active site on E2. Then the lipoic acid moves to the other active site. You get coenzyme A coming in, and you get a nucleophilic attack from this sulfhydryl on the carbonyl, and it basically just exchanges it and puts it on acetyl-CoA. Okay. How is this regulated? Well, it turns out it's regulated, the whole complex is regulated based on whether the E1 subunit is phosphorylated or not. Okay. So when E1 in pyruvate dehydrogenase complex is phosphorylated, it's inactive. Okay. And when it's dephosphorylated, it's active. Okay. So there's two regulatory elements, and they're on, I believe, on the same actual enzyme. Okay, one is a pyruvate dehydrogenase kinase, or PD kinase, that adds the phosphate groups. The other is a phosphatase, which removes the phosphate groups. Okay, so we're going to focus primarily on the kinase, what's adding the phosphate groups. Okay, when we talk about regulation here. Okay, so what turns off this kinase? Okay, so the kinase is adding phosphate groups. You can think of the phosphatase as sort of always being active. Okay, what really turns it on is magnesium and calcium ions, but that's maybe a little bit less obvious why that's a regulatory element here. So you can kind of just think of this as always being active, and the main regulation is going to come from whether or not the kinase is active. Okay, so let's focus, I guess, first on what turns the kinase on, and that are the products of pyruvate dehydrogenase. So NADH and acetyl-CoA, when those are in higher concentrations, it's telling the cell, we have enough energy, we don't need to create any more acetyl-CoA. That will turn on this kinase, it will phosphorylate E1 and turn it off. Okay, so essentially stopping the production of more acetyl-CoA. Okay. What turns the kinase off, which then turns E1 on, would be the substrates for this enzyme. So when pyruvate concentration, NAD, and coenzyme A concentrations are higher, that will turn off the kinase. Okay, the phosphatase will dominate, and E1 will be primarily dephosphorylated, and the complex will be active. Okay, and we'll see that when we talk about the citric acid cycle, this idea of substrate regulation is very it happens a lot in the citric acid cycle. There are no, at least in this class that I know of, there are no allosteric regulators of the citric acid cycle like we saw with glycolysis. Okay, so moving on to the second section of chapter 16, and this is where we talk about the details of the citric acid cycle. Okay, this is the figure the book gives. Okay, this is the citric acid cycle. Okay. This figure has a lot of detail in it, which is good. I suggest you spend some time looking through these different figures. They all have a little bit different information. It's probably hard for me to go through this in a lecture and for you to get all that information. We're going to be talking about each of these steps individually, and there are 8 steps in the citric acid cycle. Step 2 is sort of broken down into two steps, 2A and 2B. For the purposes of this class, I think just, you don't necessarily need to know that there's an intermediate called cis-aconitate. Just saying citrate goes to isocitrate is probably good enough for this class. But this figure is nice in that it shows the specific chemical structures of all of the intermediate compounds. It has in blue the enzyme names, and then it highlights in red where those carbons are going that come in from acetyl-CoA. And we'll mention this, many times, but I think I have a better figure that shows this. But the carbons that come in off acetyl-CoA are not the carbons that are lost from CO2, at least in the first cycle. Then when they go into the second cycle, those carbons, they can be lost. Okay, and you'll notice too that we stop showing the highlighted carbons at this step, and that's because succinate now is a symmetric molecule, so those carbons can be on either side. So you lose the tracking ability there. Right, here's another figure of the citric acid cycle. This is color coded, so this may be a little bit easier for you to see where what carbons are coming in and what carbons are potentially leaving here as CO2, probably a little bit more importantly. Right, so this is oxaloacetate. It's going to combine with acetyl-CoA to form citrate. Okay, the two carboxylic acid groups on oxaloacetate are what are going to be lost as CO2 in the next cycle. Okay, and the carbons from acetyl-CoA are not lost. And then finally, this is yet another way to depict the citric acid cycle. I like this one because it shows you the enzyme names are nicely highlighted here, and they give you the enzyme class numbers, so you can tell what kind of enzymes they are. So Econotase, it's class 4, so it's a lyase, similar with Citrate Synthase. And Isocitrate Dehydrogenase is class 1. It's a oxido reductant. It catalyzes oxidation reduction reactions, and so forth. Right, and you'll notice there's kind of a theme there. There's three lyases and 3 dehydrogenases. Okay, so you're doing oxidation reduction, and specifically you're oxidizing these carbons from acetyl-CoA coming in, and then you're also doing some molecular rearrangement, and we'll kind of go through each step. So hopefully it'll make a little bit more sense logically why each step is occurring. And I've heard this sort of described as easier or makes more sense than glycolysis, and that's I think that's probably sort of personal. Everyone would be a little bit different, but I think the citric acid cycle, if you look at it just as a whole, it's can be sort of confusing, but when you take each step individually, I think it does tend to make more sense. Okay. So the sequence of events, right? There are 8 steps. Okay, what's going on in general in each step? Well, the first step, we're forming a carbon-carbon bond. Okay, that's making citrate from oxaloacetate and acetyl-CoA. Okay. Step 2, we're rearranging that. We're doing an isomerization, okay? And that's via, we'll talk about this, but we do a dehydration, then a rehydration to do a parent isomerization. Okay. And what we're doing there is making a better part on that molecule to oxidize. Okay, so in step 3 and 4 is where we get the oxidative decarboxylations. We're taking off 2 carbons in the form of CO2 to give NADHs. Step 5 is where we phosphorylate a substrate to give GTP, which is, again, energy. Step 6, we do a dehydrogenation to get a reduced FADH2, an electron carrier. Step 7 is a hydration. And then step 8 is another dehydrogenation to give NADH. Okay. So let's look at these in a little bit more detail. The first step, we're taking Acetyl-CoA, that was our product from Pyruvate dehydrogenase complex, and oxaloacetate, which is a 4-carbon molecule, has carboxylic acids on the ends, each end. Okay, one middle carbon is a carbonyl, and the other middle carbon is a CH2, methylene. Okay. So this enzyme, Citrate Synthase, takes acetyl-CoA and oxaloacetate, and it makes citrate. Okay. This is a very favorable reaction. Why is that? It's because this CoA bond to the acetyl group, it's a very high energy bond. Okay, so this is a very active starting material. So it uses the energy from that release of the CoA to form this molecule, citrate. Okay. This is, and we'll mention this. I should point out the structure of citrate here first. That the acetyl group here is being added to this carbon, the one with the carbonyl on oxaloacetate. Okay, and then we get a 6-carbon molecule, which is citrate. Okay. This is the only reaction in the citric acid cycle that has a carbon-carbon bond formation. Okay. This enzyme, citrate synthase, uses acid-base catalysis. We'll see a little bit of detail to the mechanism here in a second. The carbonyl of oxaloacetate is a good electrophile, and the methyl of acetyl-CoA is a good nucleophile. Okay, so they react together. Okay, this is the rate-limiting step of the citric acid cycle. Okay, and you might see CAC, citric acid cycle. That's the abbreviation for citric acid cycle. Okay. And also, before we get too far, I wanted to mention this, but I forgot. Sometimes you hear this referred to as the Krebs cycle, and that's maybe an older term for it. But Krebs cycle and citric acid cycle, you can think of as they mean the same thing. Right. The activity of this enzyme, citrate synthase, really depends on the concentration of oxaloacetate. Okay. This is a very favorable reaction, but it is the rate-limiting step of the citric acid cycle. Okay, so why is that? Well, it's regulated by substrate availability and also product inhibition, but mainly substrate availability. And in general, the concentration of oxaloacetate is fairly low in the cell, and whenever you have oxaloacetate, as long as the products of this reaction aren't inhibiting, this reaction goes forward. Okay. This enzyme undergoes what's known as an induced fit. So there's a conformational change in this enzyme. And the first thing that binds, the first substrate that binds is oxaloacetate. Okay, oxaloacetate binds, and the citrate synthase will be in the open conformation, and only oxaloacetate can bind. Once oxaloacetate binds, it then closes into the closed conformation, and that creates the active site for acetyl-CoA. Okay? And so what this is doing is it's really making it so that it's not going to bind acetyl-CoA and maybe use it or have the chance of some side reaction occurring with acetyl-CoA unless there's oxaloacetate there to react with it. So acetyl-CoA you can think of as sort of a more precious. Acetyl-CoA can still be used to, if you don't need to create a lot of energy, it can be used in other things still. So if you don't have that oxaloacetate available, you're not going to waste an acetyl-CoA. Okay. And a picture of that would look something like this. This is the open conformation of citrate synthase. It's a dimer here with two active sites. Once oxaloacetate binds, then you can get this closed form, and this is the crystal structure of the closed form with oxaloacetate and then an acetyl-CoA analog that they put in to help crystallize this. Okay, we'll look at the mechanism of citrate synthase, and it's a general acid-base catalysis. Okay, so the first step here, we already have oxaloacetate bound, which created the closed form, have the active site for acetyl-CoA. Okay. Acetyl-CoA will come in this active site. There's an aspartic acid that then takes a proton from acetyl-CoA, and ultimately, if you follow the electrons, we have acetyl-CoA, the carbonyl here, taking a proton from histidine, which would be the base. So we have the acid and a base. Okay, and you get this Enol Intermediate. So ene meaning a carbon-carbon double bond, and ol meaning an OH group, oxygen, hydrogen, alcohol group. Okay, that's has this hydrogen bond interaction with the histidine. Okay, that's a very active compound, this enol, and then you can get, following the electrons, you can get the double bond here can attack the carbon, the carbonyl carbon in oxaloacetate. Okay, and that connects this together and forms that carbon-carbon bond. Okay. And then finally, you just get a replace. This is replaced by a water. The CoA, is replaced by a water coming in. Okay, so this CoA bond then gets replaced, and that really one of the driving forces for that energy that's released when that bond is broken. Okay, step 2 is our isomerization reaction. Okay, and we have citrate now as our substrate, and it's going to be rearranged into isocitrate. So what is going on here? Basically, we're moving an OH group from this, I guess you could call it the second carbon, this in the chain, to the first one. Okay, so you're having this OH and hydrogen are removed in the form of water, so it's a dehydration for that step. your intermediate here is cis-aconitate, and another water molecule comes in, and this time the OH group is on, a different carbon there. So that would be carbon 2 as opposed to carbon 3. Okay, so 1, 2, 3, 4, 5. You can think of this as a 5-carbon chain, and you're just moving it from, instead of the center carbon, you're moving it one out, the OH group. Okay, and that makes this this CO2 here a better leaving group when you have the OH group there. Okay, here this Econotase, how we describe this is an elimination of H2O from citrate, gives that CC double bond. Okay, that's that lyase function of the enzyme. Citrate's a tertiary alcohol. I was kind of alluding to this. It's a tertiary alcohol, which is a poor substrate for oxidation. Isocitrate is a secondary alcohol, which is a good substrate for oxidation. Okay. So you have an elimination of H2O, you get that intermediate with a carbon-carbon double bond. Then you have an addition of H2O to cis-aconitate that's stereospecific. Okay, thermodynamically this is unfavorable, and it's a reversible step. How it's made to go forward is that product concentration is kept low, so this is pulled forward. And I think that's kind of a theme here with this cycle. In this enzyme, Econotase, that catalyzes this reaction, uses iron-sulfur clusters, okay, to accomplish this. And iron-sulfur clusters, this is a 4-iron, 4-sulfur cluster. They're sensitive to oxidative stress. So oxidative stress, in times of oxidative stress, these iron-sulfur clusters can be modified and actually become inactive, which would not be a very good thing, because if you don't have the citric acid cycle, right, that cell's not gonna survive very long. Okay. Just a little aside, I've used this enzyme Econotase in a gel activity assay where you combine cis-aconic acid, isocitrate, and then a phenazone methosulfate, and then you see how active. You also use Isocitrate Dehydrogenase, right? But you're seeing how active the Econotase is by this reaction, which ultimately, when this molecule is reacted upon and reduced by isocitrate dehydrogenase, that is when you get the color change in this gel. And so you can see here, thing I wanted to point out is that there's a mitochondrial version of Econotase, and there's also a cytosolic version of Econotase. So that's one example of an enzyme that is in both the mitochondria and the cytosol. Slightly different versions, but it is in both places. Okay. And that brings up another topic here, known as moonlighting proteins. Okay, and those are proteins that have multiple roles in the cell. Okay, and it's something like GAP dehydrogenase, glycerol aldehyde 3-phosphate dehydrogenase, which we saw in glycolysis. That is a very common housekeeping or moonlighting protein that is expressed very, very highly in all cells, not just because it's important in glycolysis, but it also has activity repairing damaged DNA and in histone transcription. And because of that, you can use it when you're doing gels or Western blots. You can use it as a loading control to ensure that you're loading the same amount of cellular material onto each lane of the gel. Because if you can think about it, if you're trying to say, okay, here we have, let's just say these are two different patients. Okay, patient 5 has more of this protein than patient 6. Well, you better be sure that you're loading the same amount of cellular material or tissue on each lane to make that conclusion. And so you can use housekeeping or moonlighting proteins to do that. Okay. Back to Econotase. Econotase is, as we mentioned, stereospecific. Only the R isocitrate is produced by Econotase. And how that occurs is, if we look at the susceptible bond, the bond that we want to react on in isocitrate, right? When that interacts with the active site, it only interacts to where that bond can only be positioned in one spot. Okay, so there are complementary binding points in the active site that gives it this stereospecificity. Okay. If we look at this in a little bit more detail, isocitrate is oxidized by transfer to NAD+. Technically it can be NAD+ or NADP+, but we'll stick to NAD+. Okay. So you have this hydride transfer, so a proton and a hydride are being lost from isocitrate, okay, and that's being used to reduce NADH. Okay, then you have at this spot where you're getting an oxidation, you're going from an alcohol to a carbonyl. Okay, then you can have this rearrangement. When that occurs, this rearrangement where CO2 now can leave, right? So that's why we wanted the alcohol there to make CO2 be a better leaving group. That happens, and you get an Enol Intermediate, which then can be rearranged to form what's known as Alpha-Ketoglutarate. Okay, so this is step 3, the first oxidative decarboxylation catalyzed by the enzyme Isocitrate Dehydrogenase. Okay, so Isocitrate Dehydrogenase, we're taking isocitrate and we're producing alpha-ketoglutarate. Okay. And alpha-ketoglutarate, you can sometimes see it abbreviated as alpha-Kg. Okay, so isocitrate dehydrogenase, again, first oxidative decarboxylation, lose a carbon as CO2, and you generate an NADH. The oxidation of the alcohol to a ketone, and that transfers a hydride to NAD. Cytosolic, there's a cytosolic version of this. That's the one that uses NADP+, but we're really not concerned with that. We're going to focus on the mitochondrial version that uses NAD+. This is highly thermodynamically favorable, which makes it an irreversible step. It's regulated by product inhibition and also ATP. Okay, ATP just shows that there's a lot of energy in the cell, and you don't need to be flying through the citric acid cycle at that point. Okay. If we look closer at the mechanisms of isocitrate dehydrogenase, it's a metal ion catalysis. Okay, so that's where we get this manganese in the active site. Okay. And we kind of looked at this previously, so I won't go too much into detail, right? But you have the metal ion in the active site makes it easier for you to lose that hydride and reduce NAD+. And you're forming the carbonyl group there, which then you can carbon gets lost as CO2. And again, that carbon loss as CO2 did not come from the Acetyl-CoA that was just put into the cycle. The carbons have to stay at least one turn in the cycle. Okay. Then you get the Enol Intermediate, which can undergo a rearrangement, and that generates Alpha-Ketoglutarate. And Alpha-Ketoglutarate, if we look at this, it's a 5-carbon molecule, 1, 2, 3, 4, 5, has carboxylic acid groups on both ends, and then it's asymmetric in that there's a 1 carbonyl that's close to one of the ends, and then 2 methylene groups. Okay. Step 4 is the final oxidative decarboxylation, and that's where we have alpha-ketoglutarate, shown here in the enol form. That is oxidized, and a CO2 group is lost, and it forms Succinyl Coa. Okay. This reaction is carried out by a complex known as alpha-ketoglutarate dehydrogenase complex. And this is very similar, in fact, same exact mechanism as the pyruvate dehydrogenase complex. The only difference is the active site is specific for alpha-ketoglutarate instead of pyruvate. And if you look at the two, they're sort of similar in structure. Alpha-ketoglutarate is just a couple carbons longer. This utilizes a coenzyme A and an NAD+ as well as coenzymes. And just like the pyruvate dehydrogenase complex, this is a very thermodynamically favorable step. Okay. Again, last oxidative decarboxylation. So net a full oxidation of all carbons from glucose. When you have glucose has 6 carbons, it goes through glycolysis to make 2 pyruvates, 3 carbons each. Okay, when those 2 go through pyruvate dehydrogenase, you lose 2 CO2s. Okay, when 2 acetyl-CoAs go through the citric acid cycle, you're also going to lose 2 more CO2s from each. So you add those up, 4 plus 2, that's a total of 6 carbons that were lost. Okay. If you're following 1 acetyl-CoA, you're going to lose 4 carbons after 2 turns of the citric acid cycle. So there's some kind of record keeping involved there. Succinyl Coa is another high energy thioester. It's bound to coenzyme A. And so that generates a very high energy, another high energy intermediate. Okay. This again is very favorable step, which is irreversible. And it's regulated by product inhibition. So the buildup of Succinyl Coa, the product, will inhibit this reaction. Okay. Now, if we look at the origin of carbon atoms in the CO2s that are generated, both of these carbons, this is citrate, the product of the first step. If we look at these are the 2 carbons that are going to be added from acetyl-CoA. The carbons that we lose from the citric acid cycle are going to be those that were from the oxaloacetate originally. Okay, so when we get to Succinyl Coa, the product of this step, these 2 carbons highlighted in red on Succinyl Coa are the ones that came in from acetyl-CoA. As I mentioned, this is alpha-ketoglutarate dehydrogenase. It's a complex. you see it abbreviated as OGDC sometimes, or Alpha-KGDH. Whatever you call it, it's very similar, almost identical to pyruvate dehydrogenase complex, right? You have a TPP. That group attaches to, in this case, it's alpha-ketoglutarate, also known as 2-oxoglutarate, which is why you get the OG here, oxoglutarate. You get that attachment. That's then rearrangement occurs. You lose CO2. It's then transferred to lipoic acid. Right, lipoic acid then sticks a coenzyme A on this and produces succinyl CoA, and then you have to have a way to recycle the lipoic acid back to the oxidized form, and then you use that same sort of cycle where you have FAD being reduced and then reducing an NAD plus. And it is very similar. The gene names are shown here. The OGDH is very similar to E1. DLST is the same as the E2, and in fact, the DLD is the same enzyme as E3. As far as I know, the E3 and DLD are the same in both of these complexes. And just as an aside, this is the enzyme system I work on, which is the glycine cleavage enzyme system, and it also shares, a lot of similarities with those two complexes. You have a lipoic acid. It's a 4 protein complex instead of 3, but you have an H protein and a T protein that are sort of like an E2. A P protein, which is sort of like an E1, where you have a glycine. It takes a CO2 off a glycine, sticks it to a lipoic acid, which then gets moves that remaining part of glycine and from the lipoic acid sticks it to tetrahydrofolate. Okay, and then the L protein is recycling the lipoic acid and generating NADH. Okay. These different complexes here that have are specific for different substrates are an example of divergent evolution. So they use the same coenzymes, identical mechanisms. Their structures are very similar. The only difference is you have active sites that are different to accommodate different shaped and sized substrates. Okay, so you have in the citric acid cycle, you have the enzyme system, the active site specific for alpha-ketoglutarate. Pyruvate dehydrogenase is specific for pyruvate. And you'll see those are, somewhat similar, right? You have this acid with a ketone next to it, right? And in both of them, the size of the side chain is the difference. Okay, and then you also have another complex that's specific for the oxidation of isoleucine. And also leucine and valine kind of go through this pathway as well. Okay, so all very, very similar, using the same mechanism. Okay, moving on to step 5. Okay, step 5, we have Succinyl CoA being converted to succinate. And the enzyme that catalyzes this is Succinyl Coa Synthetase. Okay, and that enzyme name really tells you that it suggests that it's making succinyl CoA. And when it was discovered, it was probably discovered in the reverse direction. Right. The important part of this reaction is that you're generating, by removing this thioester, this high energy thioester bond, you're doing what's known as a substrate level phosphorylation. So you remove that bond, use the energy to phosphorylate a GDP into a GTP. Okay. Okay, so this is a substrate level phosphorylation. Okay, that energy of the thioester allows you to incorporate an inorganic phosphate onto GDP. Okay. It goes through a phosphoenzyme intermediate. GTP can be used to can be converted into ATP, which is why we say a GTP is equivalent to an ATP. This is slightly thermodynamically favorable, which makes it reversible since it's only slightly favorable. Product concentration is kept low to pull this forward. Okay, and the mechanism of it is shown here. We have Succinyl Coa coming in. The inorganic phosphate is actually put on to the succinyl where the CoA went. So phosphate comes in, CoA is replaced, and you get a phosphate bond directly to your substrate, succinyl. Then that is transferred to a histidine residue, okay? So and succinate leaves, and then that phosphate that's on histidine can be placed on a GDP to make GTP. So this is kind of just like a three-step process of moving an inorganic phosphate around. Okay. Step 6 is an oxidation, and its substrate is succinate, and the product is fumarate. Okay. It's catalyzed by the enzyme Succinate Dehydrogenase. So that's telling you succinate is its substrate, and it's a dehydrogenase, which is which catalyzes oxidation reduction. Okay, succinate is a 4-carbon, completely symmetric molecule, okay, which is why you lose the ability to track where each carbon came from in it. Okay. It's an alkane, so all carbon-carbon single bonds. Succinate dehydrogenase will form a double bond between the two middle carbons here. So you're removing the hydride and a proton, and you're forming a double bond, and you're reducing FAD to FADH2. Okay. Okay. Succinate dehydrogenase is different in that it's not a soluble protein in the matrix. It's actually bound to the mitochondrial inner membrane. And it's part of complex 2 in the electron transport chain. Oxidation of alkane to alkene requires FAD or FADH2. And that's because the reduction potential of NAD is too low to do this electron transfer. Okay. The FAD in succinate dehydrogenase is actually covalently bound, which is a little bit unusual. Usually FAD groups are bound with like hydrophobic interaction and hydrogen bonding. So that's a little bit unusual, but that's just really for your information. Not going to be tested on that. This is near equilibrium, which makes it a reversible reaction. And again, very kind of constant theme here. Product concentrations kept low to pull this reaction forward. Okay, so this is what we're looking at with succinate dehydrogenase. And so kind of what the enzyme we think about as succinate dehydrogenase in the citric acid cycle would actually be the A subunit. So SDHA, which is succinate dehydrogenase A, complex, not complex, subunit A. Okay. So we have succinate going to fumarate, and you're reducing FAD to FADH2. Right. This is bound to complex, what's known as complex 2. The FAD-H2 then transfers its electrons through iron sulfur clusters and ultimately to what's known as ubiquinone. Ubiquinone can then transfer those electrons when it's reduced to QH2. So Q stands for ubiquinone. QH2 is, so Q is the oxidized version, QH2 is the reduced. That then transports through the inner membrane electrons to the next complex in the electron transport chain. So we'll be seeing this again. Okay. All right. The next step, 7, we have fumarate, and it's being converted into malate. And what we're doing here is we're essentially adding water to this double bond to produce an alcohol. Okay, so fumarate is converted to malate, and specifically L-malate by the enzyme known as fumarase. Right. It's stereospecific. Okay, the addition of water is always trans and forms L-malate. OH adds to fumarate, then a proton adds to the carb anion intermediate. You can't distinguish between the inner carbons, so either can gain the OH group, right? So these it's symmetric, right? You can't distinguish between the inner carbons, so either of those can add the OH group. And that's kind of where you lose the ability here to say, that carbon, since you've lost the ability from succinate, you can't distinguish, which carbon is which, where it came from exactly. Okay. This is slightly favorable and reversible, and again, product concentrations kept low to pull the reaction forward. Right. This just shows the stereospecificity of fumarase, the enzyme, right? We have fumarate, which then makes L-malate. Right, if you have the substrate known as malleate, which is the cis version and not the trans, then you would get D-malate. Okay, the final step in the citric acid cycle, we have malate being oxidized to oxaloacetate. Okay, right, so we're oxidizing that alcohol group on malate, reducing NAD to NADH. Okay, and when that's done, we have that 4 carbon, 2 acids on each side, 1 ketone and 1 methanol, which is oxaloacetate. Okay, you'll notice that this is not very favorable, okay, which is one of the key differences between this step and the other steps in the citric acid cycle. Okay, this is what we consider the final step. Since we started with the addition of acetyl-CoA, this will be the final step. It regenerates that oxaloacetate needed for citrate synthase. It's highly unfavorable and reversible. Okay, how this is moved forward, though, is oxaloacetate concentration is kept very low by citrate synthase, which again pulls the reaction forward. Okay. So if we look at one turn of the citric acid cycle, right, we have acetyl-CoA coming in, going through one turn. We're going to create 1, 2, 3 NADHs, which is a total of 2, 4, 6 electrons from them. We're going to lose 2 carbons as CO2. We're going to generate 1 GTP, which is an ATP equivalent, and then we're going to reduce and generate 1 FADH2, which is again 2 electrons. So we have a total of 2, 4, 6, 8 electrons being generated. one ATP being formed and two CO2s leaving. Okay, so that would be our net reaction. Okay, acetyl-CoA plus 3 NAD pluses, an FAD, a GDP, and an inorganic phosphate, going to 2 CO2s, 3 NADHs, FADH2, GTP, and if you want to balance it completely, you have the coenzyme A from acetyl-CoA and 3 protons, and you needed 2 waters. Okay, not as important that you remember the waters, but knowing the other things, the reduced cofactors, the GTP and the CO2s are important. Okay. The energy captured by electron transfer to NADH and FADH2, we'll see that becomes very important. Okay. It generates 1 GTP, which can be converted to ATP, but that's not really the important part of this. It's the reduced electron carriers. Okay. And that's kind of driven home in this table. So this shows you how much ATP is either used or formed by specific steps. So we have the 2 steps in glycolysis that were priming steps where we're actually using ATP, right? So the number of ATP formed from those are -1, right? And then we have the other steps in glycolysis where we make NADH, and then when we make ATP directly, right? So we're forming through those steps, we're getting some NADH and forming some ATP. So after glycolysis, we do have a net production of ATP. Not much, but each glucose molecule, we use 2 ATPs and we make 4, so we have a net production of 2 ATPs. Plus, this can get a little confusing, but the NADHs could either be 3 or 5 ATPs formed from each of those, depending on how those get into the mitochondria. Okay, so you do have some production of ATP, and you see that NADH accounts for a lot of it from glycolysis. Okay, but going through the citric acid cycle, the pyruvate dehydrogenase complex makes 2 NADHs per glucose, which is used to produce 5 ATPs. So that one step makes more ATP than we made in glycolysis almost. Okay, and then the citric acid cycle itself, the NADH produced and the FADH produced make tons more ATP than you get from glycolysis. So that you can see that this citric acid cycle is much greater in yielding ATP than glycolysis. Okay. Another important thing about the citric acid cycle is that the intermediates can be used to produce molecules that are needed in the cell. And so that's shown here. There's many different branch points out of the citric acid cycle where you can make fatty acids or sterols with citrate. Things like alpha-ketoglutarate can be used to make glutamate, which is an amino acid. That then can be converted into other amino acids, or it can be converted into purines. Succinyl CoA can be used to make porphyrins, and a very important porphyrin is heme. Oxaloacetate can be used to make amino acids, aspartate and asparagine, and then further on to pyrimidine bases. It can also be used to create PEP, phosphoenol pyruvate, right, and then make more glucose via gluconeogenesis. PEP can also be used to make other amino acids. So a lot of these amino acids can be made by citric acid cycle intermediates, and also if you go in the reverse direction, that's how they can be metabolized too. They're metabolized back into citric acid cycle intermediates that are then fed through the citric acid cycle. Intermediates in the citric acid cycle can be used in biosynthetic pathways, and that they can be removed from the cycle. When you do that, you must replenish the intermediates in order for the cycle to continue. And 4-carbon intermediates are formed by carboxylation of 3-carbon precursors, and some of those are shown here. Things like pyruvate, that's a 3-carbon molecule. You can add a carbon to it to make oxaloacetate. Phosphoenol pyruvate, again 3-carbon. You can make add a carbon to make oxaloacetate in a couple different ways. And then those are, I should mention, those are shown here in red, where you replenish your citric acid cycle intermediates. Here are shown in red. Right, so there's a couple places where pyruvate can be put back in, either as oxaloacetate or malate, and then phosphoenol pyruvate can be converted to oxaloacetate by 2 enzymes, PEP carboxykinase or PEP carboxylase. Okay. One of the important cofactors in doing this is biotin, and as we've seen in the case of TPP or lipoic acid, biotin is also a carbon carrier, but it carries specifically CO2. Okay. And not so important that you know this mechanism, but knowing that biotin, the basic structure of it, you have these 2 5-membered rings. The important part here is these nitrogen groups. Those can be used to grab a CO2 group and transfer that CO2 group to something else. And in this case, it's you're transferring it to pyruvate to make oxaloacetate. Okay. So these are just some examples of tethers that we've seen that are used to transfer, small carbon units, right? Lipoic acid, biotin, and coenzyme A, this sulfhydryl group on coenzyme A. Okay, the last thing we'll talk about is the regulation of the citric acid cycle. And it's sort of easy because it's all driven by substrate and products. Okay, substrates activate these enzymes, and products inhibit. Okay, so really, what you'll notice is ATP is a big one, right? ATP tells you that you have enough energy. You want to inhibit this cycle. Okay, NADH also another indication of cellular energy. Okay, AMP or ADP are NADHs. Those tell you that you might be low on cellular energy when those concentrations increase. So those are activating. Okay, and also the other one that's probably not super obvious, the calcium, is another signal that turns on the citric acid cycle. Okay. Where the citric acid cycle is regulated, specifically it's regulated at the highly thermodynamically favorable or irreversible steps. Okay, which is sort of this similar to what we saw with glycolysis. Those irreversible steps are the ones that you want to regulate, and those are the pyruvate dehydrogenase or pyruvate Dehydrogenase complex E1, Citrate Synthase, okay, Isocitrate Dehydrogenase, and Alpha-Ketoglutarate Dehydrogenase. Okay. Generally, they're activated by substrate availability and inhibited by product accumulation. So just as a general rule for the citric acid cycle, you can say these enzymes are activated by substrate availability and inhibited by product accumulation. Okay, overall, the products of the pathway are NADH and ATP. So those are going to play probably the biggest role in product inhibition. Okay, you can think of those, although they weren't necessarily listed for each step there in the previous diagram. You can think of those as affecting all regulated enzymes in the cycle. Okay, NADH and ATP are the inhibitors, and the activators are NAD plus and AMP. Okay. Right. Pyruvate dehydrogenase, as we mentioned before, is also regulated by the reversible phosphorylation of the E1. Okay, when it's phosphorylated, it's inactive. When it's dephosphorylated, it's active. Okay, and that pyruvate dehydrogenase kinase and phosphorylase are part of this complex that act to regulate it. Okay, a kinase is activated by ATP. When ATP levels are high, that means you have enough energy, so you want this turned off. So PDH is phosphorylated, right? The kinase is activated at high ATP. E1's phosphorylated, and you get less production of acetyl-CoA. When there's low ATP, the kinase is less active, and the phosphorylase removes the phosphate from E1, and then you get more acetyl-CoA production, which then goes to produce more ATP. The regulation of isocitrate dehydrogenase controls citrate levels. Econotase is reversible, so citrate to isocitrate. It's a reversible step. Okay, so isocitrate dehydrogenase, this enzyme, okay, when this is regulated, right, isocitrate levels can build up. When that builds up, this is a reversible step, so you get, if you have a lot of isocitrate around, it goes back to citrate. Citrate can then be used as another building block, but it can also leave the mitochondria. When it does, it inhibits PFK, phosphofructokinase, and glycolysis. So you might remember seeing when we talked about the regulation of glycolysis, that phosphofructokinase was inhibited by citrate, and that's one of the reasons why. When you have a lot of citrate around, that means you don't need to be going through the citric acid cycle, so you don't necessarily want a ton of glucose being wasted, being turned into pyruvate and acetyl-CoA if you have enough energy, so that feeds back and it inhibits glycolysis. Citrate synthase is also inhibited by Succinyl Coa. Okay, Succinyl Coa, if you remember, is the product of alpha-ketoglutarate dehydrogenase complex. Alpha-ketoglutarate is an important branch point for amino acid metabolism, as we sort of saw. So Succinyl Coa, when you have a lot of that present, that communicates back that, okay, we don't need to be going through this cycle, so we're gonna turn off citrate synthase. Okay, because we don't necessarily need any more alpha-ketoglutarate. Okay. Right. So that's the end of chapter 16. It's a lot of information. It probably, you can't really necessarily get it all through just listening to a lecture. So I really recommend trying to spend some time looking at this cycle and looking at the steps of each step of the cycle, and that helps make a little bit more sense of it. Right. The next lecture and final lecture of the quarter, we'll be talking about the main respiratory chain, the electron transport chain, and ATP production.